So as Karen says, I kind of a part of Luca Labs, and I think it was in the pandemic actually, so it must have been about 4 years ago, Noemi Hamilton and I sat down and realised there wasn't really much that we knew about the research in leukodystrophies around the UK. So we set up what we call Luca Labs now, and it started off with kind of me and Lydia in Leeds, Noemi, who was in Sheffield but now is in New York, and Sean Sweeney. And we all did kind of things that skirted around leukodystrophy. But actually, we realised that there was probably a lot more people and a lot more researchers in the UK doing things. And so we started to kind of talk to people. Noemi's very sociable, so she spoke to a lot of people. And suddenly we realised there's so much leukodystrophy research going on in the UK. And we all really just need to get together and talk to each other and show each other what we're doing, because working together and collaborating is the best way to move forward. So here on Friday, we decided this would be a great time to get all of these researchers, these clinicians, people doing clinical trials in leukodystrophies together in a room, and there we were. This is in the other room on Friday. The sun was shining, which was lovely, but also it was a wall made of glass, which made things slightly tricky in terms of most of the audience had to wear sunglasses for half of the day, but it was lovely. And we literally spent the entire day from 9 till 5 talking about leukodystrophies and how we can improve the lives of all people with leukodystrophies. So we split it into 3. So in the morning we had a clinical session, and so we heard from Dr. Rahul Singh, who you've heard from already this weekend, about the new highly specialised white matter disease service, and how this is really important to gather information, to learn, to ensure that the individuals, you guys with leukodystrophies, are all getting the best treatment, the best service. And kind of as part of that, we do need to kind of advance the research and advance what we know about leukodystrophies and collect as much information as we can. We then heard from 3 of the neuroradiologists who are down in London, who are part of the inherited white matter disease service. Now, again, I'm a geneticist. When I look at an MRI, I have no idea what I'm looking at other than it's a brain, so I'm going to paraphrase, and I apologize profusely if I've got anything wrong. But in essence, we heard how crucial MRI is to a diagnosis of leukodystrophy. And actually now, the more that these specialists are looking at these MRIs, they're starting to see patterns between the different types of leukodystrophy. They can spot, in some cases, what gene might be the underlying cause of their leukodystrophy just from the MRI. And again, the more scans, the more of these that we look at, the more information we get. There's also the need to be child-friendly. MRIs are not the nicest machines to climb into, even if you're an adult. So for young children, this isn't an easy thing to do. So they've now got child-friendly, like mock-ups of MRIs, playing the sounds so they get used to the sounds and things like that. So it's not just about getting good pictures, but also about making it as friendly and comfortable as possible. And also some of the new tools that are coming through, so they can now look at particular chemicals while they're doing the MRI, such as lactic acid, and that kind of gives a hint that this might be something that's mitochondrial. So trying to get as much information from a single scan as possible. Now, you're going to hear this word natural history a lot, and that's because we've realized how important this is. So we really need to understand how these leukodystrophies progress, because at the moment, we don't really know that much. These are rare disorders, okay? And so you might have the question, what's going to happen in the future? And often the answer is, we don't know, okay? So we need to look and learn from all of you guys. We need to follow, track, see how these disorders progress, because we're coming up with lots of new treatments now, but in order to judge whether these treatments are working, we need to know if they're making you better compared to if you weren't on that treatment. And that's a very, very difficult judgment to make at the moment, because there's so little information out there. And so there was an example that we heard from of a child who underwent a gene replacement therapy, and actually after 2 months the child looked worse, and that was very worrying for the clinicians. And actually at 5 months they started to look better again. And when they looked into the literature, there were similar observations that 2 months in, there was a worse prognosis, but suddenly that switched back. And so that's really useful information to know. And these are only from 1 or 2 or 3 individuals who have had treatment. Told you my voice was going. But actually, if we know now in advance that for the first couple of months that this isn't going to look any better, then we can warn, the parents can be warned, the carers can be warned, and to kind of stick with the treatment rather than stopping it. And another thing is the need for biomarkers. We need to have actual ways that we can measure success. Something in the blood, something in the, something that we can track and say, this chemical's going up, which means they're improving, or this one's going down. So we need to find things that we can use to track progress. And we also heard about the importance in the bigger picture, and although these MRIs are important, we shouldn't just make a diagnosis based on that. And the examples we heard from is because some things like vitamin deficiencies can mimic leukodystrophies on an MRI. So, it could be that, a whole load of testing and treatment plans are developed from a single MRI, when actually it might just be that, it needs a vitamin replacement or, something like that. So what was really clear is there's knowledge gaps for all leukodystrophies. We do not understand everything about any leukodystrophy yet. There's still a lot to learn. But the key thing is for global collaboration. We need to work together. We need to bring the specialities together at events like this, talk to each other, and just try and compile all of this information together to learn. Because the more we understand, the more we can help. We also heard from Christina Diaz that she's an expert in mitochondria. So these are what we traditionally call the energy, the batteries of the cell. They make energy. But actually, she explained how these have many functions in cells. And, actually, it's not always the mitochondria that are not working. And, it's sometimes something very simple like, they're making an amino acid or something in the cell that we can just replace. So it's really important, again, for the genetic testing, to really understand what's going on, because a leukodystrophy isn't always obvious on what the underlying cause is. And again, this is where the Inherited White Matter Disease Service really comes in, because you should all get referred to the service, all kind of undergo genetic testing to at least try and understand what's going underneath. We also heard a lot about TUB4A, and this is the work that's going on by Adeline Vandera in the US. And they're doing, again, this natural history study, trying to understand what happens for individuals who have a TUB4A, trying to track over time how that progresses, and then looking at trying different types of therapies to see which ones can reverse that prognosis. We've also established a huge collaborative network in the US, and some of the people in this room, I think, are part of that. And it is literally just about comparing and contrasting disease progression. And what was really crucial from this is really trying to understand what the priorities are for families. So what do you want from a treatment? Okay, we might not be able to completely cure the leukodystrophy, but what aspect would you like to see improved the most? And that was really important in trying to develop a new therapy. And at least looking at the outcomes from those clinical trials. So they've established this global leukodystrophy initiative, and really it is trying to get us ready for clinical trials. What should we be looking for? What improvement should we be aiming for? What would be a good result? What would be not a good result? Now, most of the sites are in the US, because that's where the funding is, but I think there are some other sites around the world. And it is just literally collecting information, clinical information, about how the disease is progressing. And so far, they've got 3,500 families participating, so that's a pretty impressive number. And so they're able to do all sorts of different data collections, and so they're talking to families and saying, what would you like? What do you want to see from a clinical trial? And I think the top one came out as, communication. We'd like to see improved communication from someone who's under treatment, improved health, improved comfort. So those are the priorities from the kind of surveys that they've done so far. Now, obviously, there's a huge amount of work there. I'm paraphrasing this hugely, but it just goes to show how much work how far we need to go to kind of progress with kind of getting to the clinical trial stage. We then had we moved from the clinical side to more scientific talks. And for those of you who actually presented, I've kind of taken some of your slides just to make my life easier to set this up. We heard from Elisa Laurenti, and she's a specialist, a professor in blood cells. And what she's really interested in is for things like bone marrow transplants, or at least for when we take the bone marrow cells from a patient and we treat those bone marrow cells and we put them back into patients, how do we make sure that those cells that we take out of patients remain healthy while they're out of the human body? Because as soon as we take cells out of the human body, they're out of their happy place. They start to behave in a different way. So how can we maximize the chances of success by keeping those cells happy? So we treat the cells, make the cells better, put them back in. But if we're not looking after those cells, they're just going to go back in and not do anything. So how do we keep these cells happy when they're outside of the blood? And they've found a JAK inhibitor, which is a drug that's readily available. That actually, if we treat the cells with this drug outside of the body, it massively improves the number of healthy cells that remain in the lab and increases the chances of a successful graft. You may have seen the zebrafish over there yesterday. So Noemi Hamilton was talking about zebrafish, and Noemi's interested in the immune system in the brain and how when the immune cells in the brain are not working very well, that this can lead to leukodystrophies. And what her lab is trying to show is that if we replace those immune cells elsewhere in the zebrafish, they will move into the brain when they're needed. And you can see here they're all kind of lit up green and pink in these zebrafish. This is a zebrafish head. And these are nice, happy, functioning immune cells now that are in the brain. So, they're kind of immune replacement therapies that you could inject into systemically, so in the blood, and they will make their way to the brain, which is where they want to be, and hopefully start functioning normally. We heard from Triana, who is over there. Hello, Triana. About developing diagnostic tools and for peroxisome disorders. So ALD and ABCD1 is one such protein that Triana is interested in. And what was fascinating is Triana is trying to make fluorescent dyes. I say trying to make, she has made. These fluorescent dyes that within 5 minutes of putting onto the cells, she can see if that protein is working or not, which is amazing. So you can see down here, this is for PEX3, which is ABCD3. And at the top, you can see these little blue dots. And that means when she's put the dye on, after 5 minutes, this is a healthy cell. And at the bottom, there's no blue dots at all. So within 5 minutes of adding this dye to these cells, Triana can say whether that protein is working or not. Now again, completely paraphrasing here, but it's really exciting to be able to have these assays that we can do in a lab that can measure how well a gene or protein is working, especially one that's as important as something like ABCD1. So that's really exciting work that's going on. We also heard about some gene replacement therapy that's being tested for Krabbe disease, and seeing if there's a mutation in the gene for Krabbe disease, whether we can actually go in and correct that mutation in the cells inside in the body. And so this is what they're doing at UCL. This is very early work, but seeing if we can we actually edit the DNA in the cells that have a mutation to correct that mutation, and so make that gene functional again. And they're seeing some very promising results from that. And then we heard from a vigil, so Cynthia, sorry, not Cassandra, who in the US is looking at adult onset leukodystrophies and talking to patients and doing lots of surveys, and particularly wanted to know about the impact of misdiagnosis and genetic testing. And what was really stunning, I mean, this is probably, you all probably know this already, but from my perspective as a geneticist, how bad we are. Only 25% of the families studied actually had a correct diagnosis at first contact. Most of them had an initial misdiagnosis, most of them being multiple sclerosis or frontotemporal dementia. So, getting that diagnosis first is crucial, but actually having a misdiagnosis first is worse, essentially, of not having a diagnosis. And so the impact of that was discussed, and she presented a family's journey where they had six different misdiagnoses before the correct diagnosis was found. Now, this was in America, and so the genetic testing wasn't done early on, but obviously in the UK, hopefully now as part of the Inherited White Matter Disease Service, if we can get that genetic testing done early, then we might avoid situations like this where the actual correct diagnosis comes quicker than, I think it was 10 or 15 years after the first contact. So, we're nearly there. We're now into the final session. We actually heard from 4 different clinical trials that are going on. Now, again, I'm a geneticist. I'm going to paraphrase massively, and probably, if I get anything wrong, I apologize. You're going to hear about some of these later on today. So Vigil first talked about the Illuminate study. Again, this is about adult onset leukodystrophy. And what we're trying to do, what they're trying to do is test therapies, but also try and understand the natural history of that disease at the same time in parallel, because this is the only way we can really assess accurately how well a treatment is working, by understanding how much better it's making somebody if they weren't taking that treatment. So they're enrolling up to 60 individuals and following them over a 36-month period, having regular MRIs, trying to see how this disorder progresses, taking blood and doing lumbar punctures and CSF where possible to try and look for what we call biomarkers. So what proteins are going up and what chemicals are going down as the disease progresses. And if we give a treatment, do they reverse the impact of that? And so as part of that, they're also doing their phase 2 open label trial, trying 2 different doses of iluzanebart. And so that's still ongoing, is that clinical trial, as well as the natural history study, so that the results should hopefully be out in the next couple of years. You're going to hear about the Calyx study from someone far more qualified than me to talk about this, so I'm going to keep it very brief, but lariglitazone in Calyx, is, we heard from Aaron Mystery at Minorix, and there's currently 3 studies that they're looking at. They're looking at adults with AMN, with and without brain lesions. But they're looking, as part of the Nexus study, at boys with ALD and with cerebral ALD, and the impact of their treatment, their drug. And they've also got a 3rd study on compassionate use. And again, this discussion about what do we call a success and what do we not call a success came up. And although for the advanced study, they found that the primary endpoint wasn't met, there were lots of positives that came out of that. And so learning from that and going into the next study is really important. We heard from Ionis, who have got a clinical trial for Alexander disease ongoing at the moment. And again, this same kind of theme came up about what do we measure to see if this is a success or not. And in Alexander disease, we have child onset, and we also have adult onset versions. And the phenotypes, the symptoms differ quite markedly between the 2 different onset ages. So what do we measure to say this is working and this is not measuring? Because it's got to be something that's consistent in everybody. Do we measure walking, how far you can walk, eye movement, ataxia? So I think as part of theirs, they're looking at walking and how whether you can do the 10-meter walk, but it was very subjective. And the problem we have with this, and the reason why I'm going on about it, is because these outcomes define whether these clinical trials are a success or not. And so it's really important that we define what we call a good outcome and what is not a good outcome. But also we're doing this on rare disorders that we don't really understand at the same time. So it's really difficult to make a judgment call when there's quite a lot riding on it. And then finally, I think I'm still on time, just. We heard from Orchard and MLD and their clinical trial, which is fully enrolled now, but they're, and this started way back in 2010, did the phase 1 clinical trial for this. And they're now in the long-term follow-up for this study. This is going up to kind of 15 years and seeing the long-term impact of this treatment. So it's exciting. There's still a long way to go, but there's lots of positives that are coming out of these. So that really was a whistle-stop tour of Friday. I mean, and again, apologies if I've misunderstood or got something wrong. But I think there's 3 take-home messages really from all of this. one of them was, a massive thank you to Alex TLC, because you've allowed us to have this day, extra day, as part of the weekend, and it was the first opportunity we've really had to bring everyone together in a room, in person, and it's amazing how much progress and enthusiasm and motivation you get from having meetings like this. So a huge thank you to Alex TLC for letting us do that. The second thing, and that came out from a lot of these, is a thank you to the families, because without any of you guys, we can't do what we do. And we realize for things like natural history studies, that these require you to travel, to go into hospitals, to have scans, to have blood tests, and these are not easy things for anyone to do. But it is helping, and it is making a difference. So there was a huge amount of gratitude and thanks and respect for all of the families that really help us do these studies. And the third thing, I have completely forgotten what I was going to say. So if it comes back to me, I'll just shout it out in the middle of the next talk. But a huge thank you to everyone. Thank you for having us, and we're looking forward to doing it again next year. Thank you.